Hi guys, how are you? This is like take 10 of me trying to talk to you guys about Mendel's principles. So let's see how this one goes. So I just want to go over a few things before I kind of instruct you how to do Punnett squares. And we're looking at Mendel's principles. There's three of them. We're going to just talk about two today. And they're called principles and they're not called laws because there are exceptions to these rules. But at Mendel's time, at the beginning of his discovery, these were things that were holding to be true, mostly in the pea plants that he worked with. So he really lucked out kind of finding this pattern, and he didn't really find exceptions to it. But these hold true for most patterns of inheritance, that there is usually a dominant allele and a recessive allele. So the dominant allele can cover up or hide the recessive allele. So the one that's expressed, we say, is dominant. So you can see the phenotype here is red, and then it has two capital letters. The yellow cherries are lowercase a, lowercase a. And then if there is a heterozygous, the genotype is heterozygous, a capital A and a lowercase a, it's only expressing the dominant allele. So the other principle was the idea of principle of segregation, that when a parent makes sperm or eggs, their genes separate, and the gametes contain only one of the pair, or half of the information. So while right here, there may be these three pairs of chromosomes, in the possible gametes, they only have one of each, half of each of those chromosomes. Okay, so, and you can tell they're going to vary in which ones that they have. So this principle of segregation and this dominant and recessive pattern is really what's going to help us figure out what are the chances of offspring having different combinations from the parents. So this goes into a little bit of math. What's probability? Probability is the likelihood that something is going to happen, the likelihood. Observed is what actually happens. Expected is what should happen. So what Mendel did to test his theory is he would grow 1,000 pea plants and he would count what actually happened, and that was his observed. His expected is what he thought should happen. Now, if I roll a dice and there's 6 sides to a dice, nothing tricky about it, and I roll it, what are the chances that I would get a 1? Well, it's 1 out of 6. What are the chances that I'm going to get a 2? It's still 1 out of 6 chances. So is that what's going to happen though? Not necessarily. So what we expect to happen and what actually does happen, or they can differ. So just like Mendel, if he was expecting to get a 50-50 ratio, he didn't necessarily get 500 plants that were tall and 500 plants that were short. Maybe he had 400 that were short and 600 that were tall. But you always kind of look for commonalities of those ratios. So this is the rule of multiplication that we might occasionally use in genetics when you're trying to figure out what is the likelihood that 2 or more independent events are going to occur together. So for example, if you flip a coin, like what's the chances of you flipping heads twice? Well, you have 1/2, because that's your option of getting a heads, and then you multiply that by 1/2 again. So there's a 25% chance that you'll get heads twice. So you can try this out at home. So what we have is Reginald Punnett discovered and came up with a possible mechanism of how to predict results of a genetic cross. So for a monohybrid cross, mono means 1. So when we're looking at one trait, you're going to start with a 4-square Punnett square. So I'm going to ask you guys to get a piece of paper and just do this with me, where you kind of draw this 4-square. The next step is to place the alleles of the parent's gametes on the outside of the square. So for example, the cross that we're going to use is a tall plant with a short plant. Now, notice the tall plant has one capital T. That's why it's still tall. That's dominant over that recessive allele. And I have this identified as mom's alleles, and this one is dad's. So what goes on the outside of the Punnett square are the alleles of the parents' gametes. And the gametes are going to have half of the information. So here we have mom's alleles segregating. There's Mendel's principle of segregation. And then here's dad's alleles that are segregating. It does not matter which parent you put where. So this is the parental generation on the outside. The next step is to fill in the boxes. You want to just be careful and take your time and write neatly so you don't make a mistake. I recommend if there's a capital letter that you always put it first, especially in this case where you're doing dominant and recessive. So you put the large T, the lowercase t. Step 2, you fill in the bottom boxes, lowercase t. Step 3, uppercase and lowercase. Step 4, the 2 lowercase t's. So you very methodically just fill in the chart. And you should have one that looks like step 4. Now you're not quite done yet because you want to summarize the results. Just a reminder, anything with a capital T is going to appear tall. That's its phenotype. Anything with a lowercase t is going to appear short. So when we look at the results, we want to write the genotypes. That would be these letters. So how many of them are big T, little t? Well, we have 1. Oops, sorry about that. Let me go back. One screen there. So we have 1 and 2 out of the 4, or 50%, or 1/2. I don't really get concerned whether you say 50% or 1/2. Usually you're going to be answering something in multiple choice, so you'll kind of notice that in the question. And then how many are lowercase t, lowercase t? There's going to be half of them. How many are capital T, capital T? Hopefully you said 0. So 0 out of 4 is 0%. Now, phenotype is how they appear, how it physically looks. I always think of the phenotype, physical appearance. So how many of them are tall? Well, anything with a capital T. So there's 2 out of 4, or 50%, and 2 out of 4 are short. Now, in this case, the genotype and phenotype ratios match, but that's not always the case. So just a recap. Genes are the letters the organism has, like the capital T, the lowercase t, little t. And when you have 2 capitals, we say that's homozygous dominant. If it's a capital and a lowercase t, we say it's heterozygous. And if it's a capital T, lowercase t, I'm sorry, this should be little t. It looks like my autocorrect actually messed that up. So that should be a little t. Homozygous recessive. The phenotype is what it looks like. Okay, so you can see this image, tall and a short, and there's their genotypes. Cross that generation, that P generation makes an F1 generation that happens to be all tall. If you cross 2 of those heterozygous, then you will notice in the F2 generation a ratio of 3 to 1. And that tends to be a pattern that you'll see. So I like this picture because it just kind of shows that your genotype, so the actual genes, codes for what something actually looks like. Now, when working on Punnett squares or when working with genotypes, if a letter is not given, make sure you choose one that's easy to tell the difference. Like letters like A and D and B, you can really see the difference between a capital and a lowercase letter. But if you use the letters like P, S's, W's, I've even noticed C's, those can be really hard. And usually if you make a mistake, it's just because you can't tell the difference between a capital and a lowercase. All right, so get your piece of paper again. We're going to try to solve one of these Punnett squares. And this one is going to be about tongue rolling. So in humans, tongue rolling is a dominant trait. Those who cannot roll their tongues have the recessive alleles, and they have 2 of the recessive alleles. So reading carefully what the problem tells you, Bob can roll his tongue and is heterozygous. Okay, so you want to keep that in mind. Sally cannot roll her tongue. What is the probability that their firstborn will not be able to roll their tongue? So the first step, you want to make a key. Okay, you want to make a key, and in this case, they did not tell you what letter to use, but since we're talking about tongue rolling, we're going to use capital T for homozygous dominant, capital T, lowercase t for heterozygous, little t for homozygous recessive. And we're going to set up our foursquare. Out here on the outside of the Punnett square, we're going to put our parents' gametes. The next step in the process is to place the correct gametes of the parents. So we know that Bob can roll his tongue and he's heterozygous. So by looking at our key, Bob must be capital T, lowercase t. So we're going to put him here. Sally cannot roll her tongue. So we're going to put lowercase t here. The next step, you got it, we're going to fill in the squares. So double check your work. Filling in the squares. Okay, now I want you to pause for a second and see if you can figure out what the probability is that their firstborn will not be able to roll their tongue. What's the likelihood or the probability? Okay, if you answered 50%, that's correct. So what we have here is we have 2 out of the 4 will likely not be able to roll their tongue. They're only going to have one child, but what this is saying is that there's a 50% chance that that child will not be able to roll their tongue. So the next time they have a child, it doesn't mean that their next child is going to be able to roll their tongue. It's still going to be a 50% chance. Okay, so as we move forward, you're going to get a little bit better at practicing these, and you're going to come across some more challenging problems where you may have to work backwards or really try to figure out what might the parents' genotypes be. So just a quick check for understanding for you to go through these. Let's see if you know the answers. So if you need to, you can pause the video and try to write down the answers, and then I'm going to go over them. Number 1, the passing of traits from parents to offspring is known as heredity. Number 2, who was the father of genetics? It was Gregor Mendel. Number 3, genes are located on chromosomes. Number 4, every gene is made up of 2 alleles, which is letter B. Number 5, the organism's outward appearance, such as wrinkled seeds, are referred to as A, the phenotype. Okay, so we're going to pause here again, and you're going to try some of these, and then I'll go over the answers. Number 6, the letters, example, capital R, that represent the traits are referred to as the B genotype. Number 7, an organism that has 2 different alleles or letters, such as capital R, lowercase r, is B, heterozygous. Number 8, looks like I have a little typo there. An organism that has 2 of the same alleles or letters, such as capital R, is homozygous. Specifically, we say that one is homozygous dominant because it has both dominant alleles. Number 9, which of the following sets would represent Mendel's parent generation? What Mendel did is he crossed true breeding plants. So this would be letter C. He crossed ones that were completely dominant for one trait and recessive for the other to create hybrids. Number 10, when 2 different alleles occur together, such as capital R, lowercase r, the one that is expressed is A, dominant. Number 11, what is the diagram shown below called? Hopefully you're saying this is a Punnett square, Mrs. Biggins, because that's exactly what it is. And what does this letter actually represent? Okay, so these letters here are actually representing the genotypes, the DNA, the allele that is in that particular gamete. Okay, so tomorrow, you have practice problems. If you are on a roll this today and you want to go right to quizzes and try the practice problems, you can, but you have Thursday to do. I think there's like less than 15 practice problems to solve some Punnett squares. You're going to need a piece of paper. That's the best way it works. You can take it up to 2 times, and you've got to get a 65% or better. If you are struggling to understand this after you do the quizzes, or you need something reset, you need extra help, please plan to stop in my office hours to get help from me. Another option, and I did not assign this one in Edpuzzle, but it's monohybrids and the Punnett square guinea pigs. It's just a video from the Amoeba sisters, and it will kind of go through basic genetic problems again. Okay, I hope that's helpful, and I will be back with another lesson in a few days.